Okay, as Shyam mentioned, I am a senior research associate with the Center for Predictive Human Model Systems, which was established last year at Atal Incubation Centre, CCMB, in collaboration with Humane Society International India. And for the past 1 year, could everyone please mute their microphones? So, Shyam, can you please do that? Okay, thank you. So yeah, so this center was primarily established to enable more human relevant research in India. So what do I mean by that? Research has for a long time has been predominantly been based on animal models, but as we are beginning to realize more and more that animal models cannot completely recapitulate all the complexities of human biology. So, and with the advent in the past decade or so, we have several new methodologies that are based on human cells and human biology, and can help us give us more human relevant answers to the questions that we are seeking. So the center was established to how we can enable more human relevant research in India. And to do this, our main activities have been focused in 4 areas. So one, we have been working on 3 white papers for the past 1 year to understand what is the status quo of research in microphysiological systems, adverse outcome pathways, systems and computational biology in India. And this is to document both the status of research, but also to understand what are the challenges to conduct more of such research in India. And second, we've also been organizing several workshops and webinars, again, to increase awareness and for capacity building in this area. Because what we have been finding out is that many people are not aware of what are the alternative methods that can be more human-based and more relevant to humans. So we have been doing this, and this webinar series is also a part of that same initiative, where every month we have scientists from India and elsewhere who come and talk about these methodologies, so that more students and scientists in India are aware about how these methods. We also gave out a grant recently in the area of two Indian scientists to build an adverse outcome pathway in the field of cancer. So that grant has currently been awarded, and the 2 scientists are in the process of building that building the adverse outcome pathway. And apart from this, we've also been writing several reviews and popular science articles, again to increase more awareness and trying to grasp what is the research in this area and challenges in both India and globally. So with this, I would like to end, and I would ask Shyam to introduce Ikbal, who would be then carrying on the next session. Thank you.
Yeah. So thank you, Dr. Surat. So now I request Dr. Ikbal to start sharing his screen. In the meanwhile, I'll just tell a couple of words about him. So he's a doctoral student in his 5th year at the Johns Hopkins University in mechanical engineering, and his interest is on mechanobiology. So now I request Ikbal to start sharing his screen and start his presentation. Thank you.
Hi everyone. Can you see my screen?
Yeah. We can see it. Please go ahead.
Okay. Thank you, Shyam, Surat, and everyone in the organizing committee for this event. And I am extremely excited to talk to you. How many people do we have now? Is it like 50? So seems like
we have 70 people on board.
Okay. So 70 people. So quite a lot of people on a Saturday morning. It's still Friday here, Baltimore time, and it's 12:30 a.m. Okay. So that's how I'm spending my Thanksgiving weekend. Anyway, I'll today talk about about my work here in Johns Hopkins, which is related to a novel kidney on a chip device. And mostly I'll talk about understanding the mechanics of polycystic kidney disease. So as the, So as the name suggests, transepithelial fluid pumping performance of renal epithelial cells and mechanics of cystic expansion. So before I go ahead, let's try to understand briefly what kidneys do and the microphysiology of kidneys. So the kidneys are extraordinary fluid filtration units. So both kidneys have, each kidney has about 1 million nephrons, which are tubular structures that filter blood and then eventually makes urine out of it. So as the blood gets filtered, it goes through these tubes, and all the necessary and important molecules, ions and water, is reabsorbed. So just to give you a scale, both kidneys filter around 180 liters of fluid every day. So I mean, we don't even drink 180. So you can imagine how many times the blood plasma gets filtered. So at the cellular level, these tubes are made up of these monolayer of cells, and they actively transport ions and water and macromolecules from the lumen side into the interstitium side, which is the basal side. So I'll talk about it again and again. So it's good to spend some time here so that we are used to the terminology. And I'll talk a lot about sodium potassium ATPase, which is a very interesting active pump which transports sodium out of the cell and takes in potassium into the cell from the ECM, and it drives a lot of ion transport from the lumen side to the basolateral side. And we'll talk about disease versus non-disease and many other things. Another motivation is, I've talked about epithelial cells. Now that you know what epithelial cells are, so let's see what they do in a simple petri dish. So once the cells polarize, what you see here is they form active fluid-filled cystic structures. So these structures are nothing but they're a result of a lot of fluid and ions coming in from the top into the basal side of the cell, and there is nowhere to go because on the basal side you have glass. The cells are seeded on glass. So they kind of lift the epithelium and form these fluid-filled structures. It's not just these 2D, so these are human proximal tubule cells. I mean, proximal tubule is a tube inside, tube of nephron in the kidney. From a development point of view as well, you see that in a human embryo, during the very beginning, at the early stages of development, the epithelial cells forming the embryo, they pump a lot of fluid and ions into the lumen from the outside. And the reason they do it is to break out of the shell called zona pellucida and eventually implant in the uterus. So at the very beginning of life itself, we see the epithelial cells are actively pumping fluid into the lumen. So we all were at one point in time very good water pumps, and the and the efficiency or the performance of how we pumped decided the fate. So I keep using the term fluid pumping performance. So my background is mechanical engineering. So from an engineering perspective, let's try to understand what a fluid pump is. So a fluid pump or water pump is basically a device that transports water from one side to another and in turn generates a hydrostatic pressure gradient. So in pump language or pump parlance, you call it head. So the way you quantify this entire process is using this pump performance curve, where on one side you have flow rate, on the other side you have the pressure that you generate. So as you can see that if you go out into the market to buy a water pump for your house, which we all have in our houses, they will give you an option of which pump you want to buy, and you will have to take a look at the pump performance curve. And the flow rate at which the pressure at which the flow rate becomes zero is called the stall pressure or the static head. Okay, so in order to understand all that, just to give you a background, I developed a microfluidic kidney on a chip device, which is basically a porous membrane sandwiched between two microfluidic channels. And I'll go deeper into the fabrication and everything that involves in making such a device in the later half of the presentation when we do a hands-on workshop. And the cells grow fantastic very nicely in the device, and eventually they show all the physiologically relevant markers that we see in the body. So the way this device works is this is the cross section of the device. So when you seed cells, the cells polarize into distinct apical site and basal site on the membrane, and then they transport fluid from apical to basal or basal to apical, depending on the kind of cells you have. The way we quantify this flow of fluid from apical to basal side is by using this microcapillary based system. So in this microcapillary system, it's very frugal actually. So we use a camera to look at the microcapillary over time, and we measure the height of fluid in the microcapillary. So once you know the height of fluid in the microcapillary, understanding or calculating the fluid flow or the pressure gradient that is developed is quite easy. So the entire setup can be placed inside an incubator and imaged for a long time, for hours and for days. So what we are doing here essentially is macroscopy rather than microscopy. So this is a quick overview. I'll go into the data part, but just take a look at how the device functions. So you can see the cells are on the apical side, and then you add, the fluid is being pumped from the apical side to the basal side. And eventually, once the fluid fills up the basal side, it rises in the microcapillary. And using a camera setup, I can basically measure the fluid height in the microcapillary. So that's a very simple way of getting both the flow and pressure at the same time, which are very important variables in understanding the mechanics of fluid filtration. So we started off with MDCK2 cells, which are very famous dog kidney cells and are a very good model to understand fluid flow and polarization. So as you can see, the fluid is rising up in the microcapillary, and on the bottom, or let's say this plot, you see that this is the way we have quantified the fluid flow and delta P. So delta P here indicates a pressure gradient, so P basal minus P apical. And as you can see that this plot looks very similar to the mechanical water pump, the pump performance curve of the mechanical water pump. And it has features similar to that, like the equilibrium flux at which the delta pressure gradient is equal to 0, and a stall pressure, which indicates that the pressure at which the flow comes to a stall. So we wanted to know the pressure that we are measuring is good or is accurate or not. So we took the dome-based system again, and we developed a pressure sensor and inserted it into a dome to measure how much pressure do these epithelial cells develop. And we found out that the pressure that they develop is of the order of around 200 pascals, which is what we measure in the device, except that in the device we have a permeable support here, and then there is fluid flow on top and on the bottom. So what we see here is that there is a lot of similarity between mechanical water pump and a biological pump, even though they are very different in function and many other features. And the way these two behave is also different. So what's the difference basically, and why is there a common pump performance plot? So we did theoretical modeling to understand what is going on. So we started we developed a mathematical model assuming a steady state description of fluid flow. We assumed that the fluid flow happens from the apical to basal side due to the transport of an idealized charge neutral solute, and x equal to 0 basically is the basal side, x equal to L is the apical side, and all fluid flow, solute diffusion, and convection only happen in the x direction. So let's look at the equations that are involved. So the Stokes equation for water flow in the cell looks like this, and the solution to the velocity profile is that, where P is the hydrostatic pressure in the cell, mu is the viscosity, and V is the velocity of fluid flow within the cell. So once you solve this, you get this equation, and due to mass balance, you have to write the continuity equation, which is this. So what you can see here is that the average velocity is essentially independent of x. So it's a constant in x direction. And the average velocity is a function, is a linear function of the pressure gradient within the cell. So PA and PB are not pressure on the outside of the cell on the apical and basal side, but PA and PB is the pressure inside the cell on the apical. So there is a gradient which drives the velocity, fluid flow, and that's what the average velocity is. So the average velocity is nothing but the flow of fluid, the fluid that is coming in from the top and the fluid that is going out from the bottom. So basically the boundary condition is the water flux at the top and the water flux at the bottom. And the water flux can be written like that, which is basically a function of the difference in hydrostatic pressure and the osmotic pressure. So what you can see is we have a flux equation on the apical side, which is P minus P naught, which is the pressure inside minus pressure outside, and C minus C naught, pressure inside minus pressure outside, and a similar equation on the basal side as well. Now that's all the equation about water flow. But we know that the water flow is driven by solute flow, and in this case we consider it as charge neutral idealized solute. So so this is the diffusion equation for the solute flux, and the flux is basically a constant, and the boundary condition dictates that we have flux on the apical side and we have flux on the basal side, and this flux of ion is composed of two different fluxes. One is the passive flux on the apical side and passive flux on the basal side, and active flux on the apical and active flux on the basal side. Now the passive flux is very straightforward because it depends on the concentration gradient, but the active flux is where interesting things happen. It depends on the cell, it depends on the kind of ion channels that the cells express. So now this model gives us the scope of examining different kinds of active fluxes. So when we use an active flux equation, which is basically a solute pump where C active depends on the concentration gradient and has a stall flux, we sort of get pump performance curves like this, where you can see that the flow rate is dropping with increase in hydrostatic pressure. One interesting prediction that we can see here is that as we decrease the osmolarity of the fluid on the apical side, the equilibrium flux and the stall pressure both increase. And we observed this experimentally as well. So if you focus on the red and the black curve here, so red is control when the cells are just pumping fluid from apical to basal side and they stop at around 150 Pascal. If you decrease the osmolarity of the apical solution by around 50%, then you see an increase in equilibrium flux and the stall pressure. So that's interesting, and that sort of tells us how is the cell behaving and why do we get a pump performance curve. Now let's move into a disease that we are interested in. We started to investigate polycystic kidney disease. This disease is a genetically inherited disease. So it happens due to mutation in two genes, which are PKD1 and PKD2. So as you can see, the disease is characterized by numerous fluid-filled cysts inside the kidney and the interstitium. So the tubules that you see, they dilate, and eventually you have plenty of fluid-filled cysts, which eventually render the kidney dysfunctional. So we have amazing collaborators from University of Maryland, so who were able to give us both cells from normal human kidneys and from the diseased kidneys, people who had to discard their kidneys due to the disease. And the disease, basically the disease cells that we got were primarily PKD1 mutation. So as you can see, in case of the normal cells, the pump performance curve is in the is as usual, like this is what you expect, because the cells are absorbing fluid from the apical to basal side, creating more pressure on the basal side, and this is how the fluid flux changes. But when you come to disease cell, you suddenly realize that the fluid flux is reversed. So the cells are now pumping fluid in the opposite direction, which is what they do when they want to form a cyst. So if you remember the previous slide, in case of a normal tube, cells basically absorb fluid from the lumen into the interstitium. However, in case of the disease cells, they form cysts, and cysts, instead of absorbing, they secrete a lot of fluid into the lumen. So the entire pump performance curve changed. The reason you have delta P negative is because now the apical side has more pressure than the basal side. So, I mean, last year, FDA approved the first drug to tackle this disease after a long time. This drug is called Tolveptin. And what they did, or what their readout was, they looked at the kidney volume or the glomerular filtration rate, which is a way to quantify how much the cells are, how much the kidneys are filtering. So they found out that the increase in size of the kidneys was lesser when the patients had tolveptin. So we wanted to see if at all the cells secrete less when it comes to tolveptin. So as you can see that in case of ADPKD control, and when you add tolveptin to the cells, the overall pump performance curve drops, and then you have less equilibrium flux, and the stall pressure that you are generated, that gets generated, is also smaller than what you have. Sorry, I mean, this scale is not right, because it should be negative 400, because they are pumping on the other side. So we wanted to get a better model for the to model the disease. What we did is we took mouse cell lines, and it's a very interesting cell type, because it's a Cre-based system where you can grow these cells at 33 Celsius with interferon gamma, like normal cell lines, and then you can differentiate them into primary cell-like behavior when you remove interferon gamma, and using doxycycline you can knock out the gene of interest. So here we knocked out PKD2 to model the disease. One interesting observation is that we looked at the barrier strength of the epithelium in the device, and we found out that when you knock out the gene, the barrier strength goes way higher as compared to the normal cells, indicating that the cells have much stronger capacity to withstand pressure in the lumen. As you can imagine, the cystic cells they grow larger and larger, and they accumulate a lot of fluid and pressure. When it comes to pump performance curve, the pump performance curve dropped with the knockout, but we did not see a reversal in fluid flow. The fluid flow was still apical to basal, but overall there was a drop in the performance. So with that, I would like to conclude this part of the story by giving you a schematic. So what we see is that in case of a normal kidney tubule, you have fluid flow. The fluid is being reabsorbed from the lumen to the interstitium. And when you generate a hydraulic pressure gradient, the tube experiences a force in the direction of the lumen, which sort of maintains the right tubular dimension. But when it comes to disease, some of the cells might have poor pump performance, or could even reverse the fluid flow from the interstitium to the lumen. And then you can see that there can be an imbalance in force along the epithelium, and that could potentially lead to dilation of the tubule. And in case of a cyst, I mean, all the fluid is coming in from outside to inside, and the forces experienced by the cell is outwards in nature. So that could easily lead to expansion of cystic. So that is basically pressure based expansion of cyst. Now, the key takeaway is that kidney epithelial cells are active mechanobiological fluid pumps, but there is an inverse relationship that you have seen with J and delta P, and we wanted to know why is that. So one interesting drug is VOBINE, which blocks sodium potassium ATPase, and I said I will talk about that because this pump drives a lot of iron and then water and helps in reabsorption. So with VOBINE, when you block NAK, you see that there is a systematic drop in pump performance by the cells. We looked at domes to quantify that. So we got a construct from the Kaplan lab in Yale, and using that we could image NAK in MDCK in a live cell fashion. So what we did is we used the kidney on a chip device to mimic what happens when the cells pump fluid and when they don't pump fluid. So delta P equal to 0 indicates when the cells are not pumping fluid. So as you can see, the NAK is more or less doing its usual thing. Nothing really interesting happens here. But when you apply pressure on the basal side, as if the cells are pumping and generating the pressure, there is a drop in NAK on the basolateral side, but when you remove the pressure, NAK goes back to the basolateral side as if like it is reversible in nature. So we quantified the entire process, and as you can see that NAK intensity on the lateral side dropped when you apply the pressure. We also wanted to look at F-actin, which makes up the cytoskeleton, and it is actively linked to NAK via tetramer through spectrin and chirin. And we did the same experiment, and it is very interesting that the cells don't do much when there is no pressure gradient, but when there is a pressure gradient, you see these high frequency invaginations going on. So I can run it again if you missed the video. So these high frequency invaginations happen when you apply pressure due to the imbalance in forces, and then when you remove it, the cells go back to normal again. So the time scale of these invagination is on the order of 20 seconds. So this sort of should tell you that this is sort of the time scale of activating the contractile machinery of the cells, because they are trying to push back the invagination. So you see the circles are diminishing. So which is, I mean, one slice of the cell is what you are looking at, and what you see is the invaginations are being actively pushed out by the basolateral cortex. And one interesting takeaway from this is that under pressure, the cells experience a disruption in F-actin cortex, which leads to a disruption in NAK that we have seen in the previous slide, because they are tightly bound. So this is, I think, the first observation of NAK ATPase being disrupted by mechanical force. So we also looked at, I mean, the primary cells again from human and mouse, and wanted to see what happens to the NAK. So if you focus here in wild type M, which is the normal cell from the medulla, and ADPKD, you see that under pressure, the NAK does not change much with disease, but under pressure in case of normal cells, the NAK intensity goes down. So clearly the disease cells have a poor understanding of pressure, and they are not behaving the same way they do like normal cells do. So we looked at the RNA content. So just, I mean, I ask you to focus on the levels within the green bar. So as you can see, the RNA levels drop under hydrostatic pressure gradient, but in case of disease, they don't necessarily drop. So similar levels were similar results we got from the mouse cells as well, where you can see that when you take the DMSO control, the RNA levels drop significantly with pressure, but when it comes to the disease cell again, PKD2 knockout, they don't. Rather, sometimes we have seen that in some of the repeats, they are of the same level as control, or they go up a little bit. So with this, I think I can draw the conclusion safely that kidney epithelial cells are active mechanobiological fluid pumps. Epicobasal fluid flux reverses in ADPKD cells. We did not see a clear reversal in case of PKD2 knockout. Delta P decreases epicobasal flux by inhibiting sodium-potassium localization, and that happens by disruption of F-actin cortex. With that, I would like to thank my collaborators and my fantastic undergrads who work with me very rigorously, and obviously my advisor, Professor Sean Sun. I will take questions now, and then I'll move into the workshop part. So I'll unshare my screen, and I can take a look at the questions that you have posted. Okay, is anyone going to help me with the question, or should I just read and answer the questions?
Yeah, I'll just maybe just go through the questions, and then maybe
we have a workshop session, so I can answer some of the questions.
Yeah, maybe we'll just take 2 or 3 questions right now, and the rest we can just take towards the end of the session. Is that okay?
Okay. Yeah, no problem.
Yeah. Okay, so the first question is by Manish Gore, who's asking what is the membrane made of, what is the pore size, and whether the medium is continuously perfused through the device.
Yeah, so yes, answer to the question, I mean, the last question, obviously the media is perfused through the device as the cells are growing, forming an epithelium, they mature, eventually form tight junctions, and then they start pumping. That takes time, at least 4 to 5 days post-confluence. The pore size is 1 micron, the membrane material is polycarbonate, and I'll talk more about the device dimension, device design, and materials I have used for the porous membrane in the workshop. I'll go deeper into the device design part.
Okay, the next question is by Dr. Brinda. She's asking were human kidney cells used in any of these studies? If not, is it because of availability or any other reason?
No. I mean, human kidney cells were used actually. So hold on. So this data, yeah, this data is all human kidney cells. Yeah. So this data is human kidney cells, and these are, I mean, kidneys that were nephrectomized from both normal patients and patients who had the disease. The disease cells are cystic cells, so they are essentially completely diseased cells. Now, this is, I mean, the disease happened in those patients because of a PKD1 mutation, but we model the disease with mouse cells with a clear PKD2 knockout.
Iqbal, there's another question from Shreya Sen Gupta. Does the chip mimic the lumen system in the kidney? For instance, are there 3D lumen present?
Yes, so the chip essentially mimics the fluid flow behavior that happens in the kidney tubules. So I'll talk more about why we have a flat uh, epithelium, as because inside the tubule you don't have a flat epithelium, you have tubes, right? And a tubular structure. And is it okay to have a flat epithelium? I'll talk more about that in the workshop part, explaining function-based design rather than just mimicking what is in the body. So what I'm trying to say is that using this device, you can get readouts that were previously not possible, like getting the flow, fluid flow and pressure at the same time. And why do we need these readouts? The reason is that once you have these downstream readouts, you can then do experiments like this, as you can see on your screen, where you can essentially see which drugs decrease the fluid flow, because eventually the kidneys have to be discarded because of excessive secretion into the lumen. So because the research is going on for about 20, 30 years, when people discovered why the disease happens. It happens because of PKD1 and PKD2 gene, but it's still unclear why do the cells secrete, and it's $1 million question in this disease. I mean, it's not simply because of NAK imbalance or some other ion imbalance, even though there is overexpression of some ions which are more ion channels which are more in the disease, but there are very interesting mechanical readouts that one should look at, and this device enables those readouts.
Yeah. There's another question from Prasun Avasti. My question is that you showed one graph between TEER OHM/CM2 versus the time. What exactly is this, and how did you measure this? So if you could just show the slide.
About,
yeah, this is the one. Yeah.
Thanks for asking this question.
Yeah.
This is the plot you are talking about, right?
Yes,
so,
yes.
This plot was, I mean, this measurement is the measurement of the barrier strength of the epithelium. So you can imagine, let me show where I can find an epithelium. So you can imagine the cells are forming, I mean, the tubes are made up of this monolayer of cells, right? So these cells have junctions, right? And they form different kinds of junctions. Some of them are adherent, called adherent junctions, gap junctions, tight junctions. So the tight junctions are crucial in maintaining the barrier property. And these tight junctions are maintained everywhere there is a barrier. In retinal epithelium, you see this barrier. In blood-brain barrier, you see this tight junctions. So using TER, what you measure is the resistance across the epithelium, and it is an indication of how strong the tight junctions are. So we measured it using a device, and I'll talk about frugal methods of measuring, but this is what the device looks like. It's quite an expensive device, but I will talk about different ways you can measure TER in an organ-on-a-chip setup. This device is basically for transwell filters.
Okay. There's another question from Tribit Kumar Bhaumik. His question is, what is the density of the sodium or potassium ATPase pump in the lumen side and basolateral side? How is it mimicked in the model? And how the cross current flow in the kidney mimicked in the microchip model?
Yes. So as you can see inside the device, the NAK ATPase are polarized on the basolateral domain, just as you would expect them inside the body. one plot that I want to show is this one. So one interesting fact about this disease is that if you look at the non-cystic tube, right? So this is a non-cystic tube. It's a cross section of a tube basically. It could be proximal or distal tube. You see that the NAK localization is on the basolateral side. So this is lumen, this is outside, right? So it's on the basolateral side. In case of cystic cysts as well, you see the NAK localization is again on the basolateral side, right? So this is very interesting. So this indicates that the fluid flow should happen from lumen to the interstitium, but that doesn't happen in case of cyst. So inside the device, I have shown it multiple times, and the localization is similar in both cystic and non-cystic cells, but the fluid flow direction is different.
Yeah. There's the last two questions. So one is, how essential is it to show TEER? So that's
not very essential. Our primary focus is is basically the pump performance curve. I happen to look at the TER plot. I always do some extra experiment that are primarily based out of curiosity. So I found something very interesting, as you can see, that the TER values totally diverged in case of the knockout.
Yeah. So there's one last question. It says from iPhone, but the question is, where epithelial cell tissues culture on the microfluidic membrane, where epithelial cells used and they used in the membrane.
As you can see in this one, the cells were plated, seated in the channel on the apical side, and the cells grow really well on top of the membrane. The dots that you see are basically pores on the membrane, and the cells, as you can see, are growing. So
yeah. So there's another one. We'll just take it. How did you perform immunos and image the cells when they were in the device?
So I have two versions of the device, and I'll talk more in detail about. So basically, this device, as you can see, has two channels, one on the top and one on the bottom of the epithelium, right? Trying to get the apical side and basal side. So depending on what kind of experiment that you want to perform, if you are more interested in imaging the cells, then you'd want to have a thinner basal channel, right? Because of working issues with working distance of a microscope. So I'll talk more in detail in the workshop.
Yeah. So there's one last question. It's from Sandra from the European Commission from the JRC. So she says, thank you a lot for the marvelous talk with a great OOC tool to study the disease. Ideal to look at therapeutic interventions, in vitro science, in vitro. Since the kidney is in the human body water pump using the novel diamagnetism therapeutic intervention, might be interesting to explore your tool similarly with COVID-19 and lung fibrosis. So that's a comment rather than a question. So if you could just share your views on whether this model can be used to study lung fibrosis and COVID-19.
Yes, of course. I mean, the device is you can always see the lung cells on the apical side and then endothelial cells on the basal side, and that's fairly straightforward. And you can model then a lung. You can make it a lung on a chip as well. You can, depending on what kind of cells. So depending on what you want to model. And I totally agree with Sandra that this can be used, because in lung in lungs you have fluid accumulation. It's called pulmonary edema, right? So where fluid gets accumulated. And there is a very interesting study, I forgot, probably from Yale University, where they showed that if you overexpress NAK, the fluid gets reabsorbed back. So you can sort of mitigate the issues with edema. And I am fairly certain that in this device, you can quantify how much you overexpress and how much of the fluid flow happens. So yeah, thanks a lot for the question.
Yeah, so thank you, Ikbal, for the interesting session on how to use kidney on a chip to understand polycystic kidney disease. So let's move on to the next session. So this is a session on a virtual demonstration of how an organ on a chip can be used using Frugal methods, the design interventions. So Ikbal has got this, I suppose, from Manu Prakash from MIT and the Frugal Science group. So so we are glad to see the demonstration right now. So the floor is up to you.
Thank you. Yeah, so I have some questions about before going into this session. Yeah, I mean, Manu does fantastic work, and he has inspired me. But this device and the idea of frugal organ on a chip is sort of something that I've been thinking about. And eventually, I happened to meet him through the class, and I told him about the idea, and he got all excited. But I mean, we'll talk about that later. Before going into the session, I would like to ask, as we have sent in the mentioned in the emails invitation, do people have some basic stationary item around them? I'm about to go into the workshop part, so it would be fantastic if you can participate. So let's make it a little hands-on. Uh, uh, so if you have, it's great. If you don't have, you can just see what I'm doing in front of the camera. I will go back and forth. You can raise your thumb virtually or just raise your hand if you have it. And you can share your uh, like
Maybe you can just type in the
unmute.
chat box if anyone has it. Maybe they can just type in the chat box, whichever is more convenient for them.
Yeah. It's not necessary, but what we'll do right now is I'll show you some tricks and tools of how to make these devices, and it's basically playing like a kid. All the things that I have here, you probably it's easy to get it from. So you can. Yeah.
Just another thing, if people have the required equipments with them, they can just turn on their video, but not the audio, and then they can just
One housekeeping rule is that please keep your
audio off. But yeah, you're welcome to
all right, I'm
turn on
gonna
the
go
video.
forward.
Yeah.
We are sort of running out of time. Let me share my screen. Hmm. Can you guys see my screen?
Yes.
Yeah.
Hello?
Yeah, we can see your
Okay,
screen. Yeah.
great. So this session is called Make Your Organ Chip. Now, the thing is, we'll talk about basically organ chips versus organoids. Those two are the things that are like kind of the buzzword these days. We'll talk about key design considerations, fabrication strategies, which are frugal but functional, which is what sort of is the mantra here. Um, uh, we'll talk about sterilization because I mean, at the very get-go, I mentioned that we are not going to use a clean room. We are not going to use lithography, and still we would be able to make fully functional organ-on-a-chip devices. But so eventually we'll need to sterilize them because eventually cells are going to get into the devices. One assumption that I'm making is that I'm assuming that everyone who is participating right now have access to a cell culture facility or maybe a microscope, so that I mean, if you eventually the device is nothing if you don't have access to cells. You'd have to put cells inside, right? So we are not going into the cell part now, but only design and device lab, and we'll do hands-on demo. So I'll go back and forth between sharing my screen and changing the camera, changing to the camera. Okay, so one important differentiation that we should understand right now is that what's the difference between organ on a chip and organoids? So organ chips are basically tools to study and manipulate cells in a culture using microfluidics and different kinds of fluidic microchannels and all these things. Organoids are, on the other hand, are self-organized in vitro tissue. So you seed some cells, you give them a cocktail of nutrients, and they grow into a specific type of a structure, a 3D structure that sometimes mimics early organ development. So as you can see on the right-hand side, you see that this is a schematic of an intestinal organoid, which forms this crypts, which is what we have in our guts. So there are stem cell crypts, and then there are differentiated epithelial cells which absorb all the glucose and ions and water that we eat and drink. However, in case of an organ chip, as you can see that we have microchannels and we seed them, they do eventually differentiate and self-organize into epithelial intestinal crypts. But there is much more control from an engineering point of view in terms of fluid flow, in terms of extracting the media and nutrients from the apical side and basal side, in terms of stretching them, in terms of plenty of, like, from an engineering point of view, we want to quantify stuff more and more, right? So if I can just pick it up and stretch it a little bit, I get to know so much information out, which is not so easy with the organoid setup. So there is less control there. There you can basically now connect multiple organs. So you have a kidney on a chip and you want to connect it with liver to understand metabolism and filtration. You can do that. You can connect both of them in serial and observe that, which is sort of difficult to do in organoids. But still, I guess people are trying to make organoids on a chip and many different things. So that's basically the fundamental difference of organ chips and organoids. Okay, so we should not confuse that. And not to say that one is better than the other, depends on what you want to observe. Okay, so if you are interested more into controlling the transport properties, you should rather be working with an organ chip rather than organoids. Okay, so and there are so many different kinds of organ chips out there right these days. So this is a kidney on a chip that we developed. There is a lung on a chip that Don Ingber developed, and then there's gut on a chip. Blood-brain barrier is very interesting. There is retinal epithelial barrier as well. People study for different diseases. This is my personal favorite. It's a blinking eye on a chip. So there is a mechanical actuator that mimics the blinking of the eyelids, on top of a layer, just to see how hydration changes with disease. And then you can, as I said, connect all of them and make a body on a chip to understand metabolism and filtration and so many drug toxicity as well. Yeah, so there is a lot of chaos out there, plenty of things, but all of them share one fundamental feature, is that there is a piece of epithelium and it has and then you can manipulate the fluid flow on top and on the bottom of the thing. So why do we want to make it frugal? The reason is not very difficult to understand. Science these days, I think, has become an arms race where you need a lot of money to do fancy experiments, and a lot of these labs don't have even a clean room, let's say, to do these experiments to make microfluidic channels and all. So what I think is important is to understand and different ways how you can make scientific investigation easier for everyone. So we'll talk about the design of the channels first. And the design is very important, not from just a design point of view, but from a functional point of view. I mean, I'm not going to design it just randomly. So the first question that one should ask is, what are we trying to model? So let's take a case of kidney on a chip. So we want to model the kidney, but what? Are we going to make a whole kidney? Are we going to model, make a nephron, which is a complex tube of cells, made up of cells, which are very different in nature. Like the proximal tubule cells are different from loop of Henle and collecting duct and distal tubule. They also perform different functions. They don't even have the same kind of ion expression and ion absorption behavior. So you have seen this picture. So one interesting fact is that most of these cells are lined up in the form of a tube, right? So as you can see, that different tubes have different features, and I'm not going to go into the detail, but what you can see is that we can cut open this tube and put it flat on a substrate and study the filtration property. So the apical to basal side filtration and ion absorption or water flow should not depend on the, I mean, unless the curvature is very small, should not usually depend. So what we are looking at is, we want something like this, very straightforward. We want to have a small piece of epithelium and have control of the fluid on the apical side and basal side. right? So that's from a design point of view. Now, so we sort of can settle that, well, we want an epithelium, and that must differentiate to form an apical and basal site. And eventually we want to have control on what ions and what water and molecules, everything is get absorbed. So at least we settled on that we need a device like this where we can seed cells, they will self-organize into an epithelial monolayer, and then there should be a permeable support, and one on the apical side, one on the basal side. Okay, so now the question is, once you know that we need a rectangular chamber like that, let's talk about the dimension of the chamber. Is it going to be a square? Is it going to be a rectangle? If rectangle, then what is the aspect ratio, as in like the height versus width, and why is it important? We'll talk about that. Is it going to be like this or like that? I mean, these designs are not random, and we'll understand as we go deeper into the workshop. One important point is that when you have a channel like that, you obviously are not going to keep the channel as it is. You are going to flow fluid, as someone asked, am I perfusing the channel with fluid? So obviously you want to grow the cells under some kind of flow condition as you would have them in the body. There is always some fluid flow in the lumen, and there is always some pressure and everything on the interstitial side versus lumen side. So it is important to consider what are the implication of the fluid flow is on the cells. So I mean, this is very basic fluid mechanics, where due to a no slip boundary condition on the edges of the wall, the fluid flow takes a parabolic flow profile, right? So that causes some problems. So as you can see, the expression here is an expression of shear stress. So you are trying to flow fluid on the top of the cells, that creates shear stress on the cells, and the shear stress is going to change because it is a stress, right? You are applying force on the cells, both normal and both tangential. So that is going to change how the cells behave. Now you have a rectangular channel, not a circular channel, because you wanted to simplify the entire process and not go into the complexity of having a circular channel. Now, the moment you have a rectangular channel, you lose the circular symmetry. So when you lose the circular symmetry, the shear stress is not going to be uniform across the circumference. I mean, it is going to change across the width of the channel. So what you can do, as you can see in this expression, I mean, this is just an infinite summation series, so don't bother too much about it. What you can see is that if you can decrease the aspect ratio, like if you can have a wider channel but thinner channel, then the shear stress expression eventually simplifies to this, and you have a much more uniform flat velocity profile, where most of the cells along the width of the channel are going to experience similar shear stress. And this is basically a quantification of fluid flow with different aspect ratio. So, okay, so we sort of removed all the other dimensions because we realized that we need to have a rectangle where the height is smaller or significantly smaller than the width. Okay. Now let's understand fabrication. What I can do is, let me see. I'm going to change the view. I hope you can see my, let me unshare for a minute. Just give me a second, please. You can pin on the screen that I have, and that way you can see what I'm going to do here. And I'll share my screen on and off. Okay. Where am I? Okay. So from a fabrication point of view, what we need is, as far as we understand, there are two channels that we need to make, and there is a porous membrane. Okay. And we eventually need to glue these two channels and sandwich the porous membrane between the two channels. Now let's start to target one channel at a time. So let's talk about the top channel. So the usual way that people make
these micro channels, and the dimensions are fairly small. We are talking about microns, 200, 110 micron, on the order of that. So due to the small size of these channels, people usually go for soft lithography. So this method, I'm not going to go deeper into it because we are not, the reason we are having this workshop is to find a way so that we can do away with soft lithography. So this method is very interesting and also very precise. With this, you can make patterns that have a high resolution, both X, Y, and Z, and then you can pour PDMS on the patterns. And when you remove the PDMS, you get the inverse pattern that you want to do. So if you wanted wells in your PDMS, you would make the inverse of the wells on a flat substrate. Most of the time, it is silicon wafer. So this method is very expensive, and you need a clean room setup to do the entire set. I'm assuming that you all understand what PDMS is. If not, I can quickly explain. PDMS is a polymer that is biocompatible. You can mix part A and part B, and after some time it cures. It has excellent properties in terms of making fine channels. So once it solidifies, this is basically a PDMS layer. So it was not solid like that. Initially, it was liquid, and it basically polymerized under certain condition. So, okay. So coming back to aspect ratio, let's fix our aspect ratio to 0.1, because that plot showed us that 0.1 is a good aspect ratio when it comes to shear stress and fluid flow profile. Now, you can see that we can keep the same aspect ratio and gradually increase the width of the channel that we are talking about, right? Because the width does not really matter that much in this case. So if you increase the width of the channel, then you don't have to necessarily rely on soft lithography. Now you can do 3D printing, which is very cheap these days. You can do machining. Almost all institutes have a lathe machine and a machining facility, and you can also outsource it, right? So once you go into a little wider channel, to start designing a wider channel, then you realize that the height doesn't have to be that much. And I would like to show you. So this is basically a 3D printed mold that many people do. And this is the mold that I've been working with. It's an aluminum mold that I have machined in Hopkins. So what you can see are these tiny structures. I hope you can see them. These tiny structures, wait a minute. Yeah, these tiny structures are basically the inverse of what I want to pattern on my PDMS. So I poured PDMS, and PDMS has become solid in this mold. Okay. So once I remove the PDMS out of that mold, you see that the patterns are now imprinted on the PDMS, but in the inverse direction. So now I have wells in PDMS. So this design is specific to my kidney on a chip, but you can have your own patterns and design. So it gives you complete freedom of what you want to design, how you want to design, right? So I mean, I'm not sure if you can see it, but there are like wells. You just have to trust me on that. Okay. Now what you can do is, I mean, this aluminum mold gave me 9 PDMS tops, right? So 9 wells. So I can now quickly take, wait a minute. So I can grab a knife. So everything we'll use here are basic kitchen and stationery items, okay? And let me wear gloves. And eventually, I know a lot of you are concerned about sterilization and you know, contamination. I will talk about that. So usually I don't do it like this with bare hands. So yeah. So you can cut it open. Make a channel like this. So that's basically one channel that you can use to make a PDMS top block. The reason and also you want to add ports so that you can add fluid in and out of the channel. So you can use a blunt tip like this, and you can just make. I mean, if something is not visible or there is a focus issue, you can unmute yourself and let me know. Okay, let's try to make it very interactive. So
yeah, Ikbal, I think there is some reflection that is falling on the thing. So it's some places it's not, sometimes not visible. So
okay.
Yeah.
hold on. Let me. Is it better now? I decrease the light intensity.
Yes, I mean, yeah, overall it's clear, but I think, yeah, I think we can move on if,
Yeah, please let me know. I mean, don't worry about interruption. Yeah. So now you have a port, as you can see. So this port can be used to, you can make two of them. Let's make two. So suddenly what you have is a top channel, right? And you can add fluid flow using, let's say, this blunt tip. You can connect a syringe pump to it, or use gravity-based fluid flow. So now it can sit on top of any substrate and create at least a channel. So as you can see, I developed a microfluidic channel simply by using a mold. And the channel is good enough to seed any cells, any kind of cells, and not disturb the cells with extraordinary forces, or I mean, the entire setup is laminar due to the aspect ratio. Okay. Now let's see. Okay, so that's fine. Now the question is of the bottom channel. So the bottom channel is very important from a point of view of working distance, because a lot of you want to image the cells sitting on the membrane. And I'm thinking, should I go into the chat and look at the questions, or should I keep going?
Someone has just said it's easier to view if you shift to camera mode, remove the presentation mode.
Yeah. Is this better?
No, I think the size is still like small, but we can't see a presentation
you can
now.
increase the size by pinning my view. So I mean, it's you can double click on.
Okay. Yeah, maybe we can just, if you just right click, this for all the participants here, if you just right click and there's a pin video option, like if you click that, you can kind of maximize the screen.
So you can maximize the screen.
Yeah, I think this is better. Yeah.
Better now? Okay. So I mean, let me just quickly recap. So this is the channel that I just made, and I created two ports in the channel. And as you can see that I can add something like this and start fluid flow within the channel. So that's basically the top part of the channel. Okay. Now the bottom side is important, because the bottom side has to be thin. So if you have a microscope with a large working distance, you can use this as the bottom side and sandwich a membrane, porous membrane. We'll talk about that between the two channels, and then use that. But it's very thick, as you can see, and it will be super hard to image that. And yeah, someone wrote about oxygen plasma bonding. We'll talk about that. And yes, oxygen plasma bonding is a way to bond, and there are other ways of bonding, because the plasma machine is $5,000. So yeah, we'll talk about all the ways you can make it frugal in nature. Okay, I need to quickly look at my presentation to see what's the next slide.
Yeah, there's one more question by Subhadra. She's asking, will the device made with this device have good resolution in comparison to soft lithography method?
Yeah, as I said, question is, what is the resolution you are talking about? So are you, do you want to make a device which is functional in terms of looking, let's say I am interested in fluid filtration, right? So I don't care if the device is 100 micron by 10 micron, or the device is 50 micron by 200 micron. All I care about is the fluid flow profile should follow a certain pattern, so that I don't stress out the cells while I'm measuring the fluid flow, right? So again, so this question again goes to the fact, points towards the fact that what happens to the basal side. So let me share my screen again just to show you something. Okay. So as I said, I changed the basal side a little bit. So the basal, I mean, these two parts were connected, right? So you could imagine that the apical side is opposite of the basal side. But as I said, the apical side is pretty thick. I mean the top side. I should not use apical and basal. So the top side and the bottom side. So what I did is I changed the bottom side to have something like this. So now I have 4 parts instead of 3 parts. Previously, I was talking about combining 3 parts and making the organ on a chip. Now I made a very flat channel on something that I'll talk about. And then on the base, I have glass or plastic. So what is that? I will unshare again so that you can see my screen. So please.
Akshay is commenting oxygen plasma bonding doesn't stay for a long time and later the 2 membranes separate.
Yeah, so we'll address that as well. Yeah, so we have not gone into bonding, by the way. So we'll talk about how do we bond the equipments. Let me see. Now I can see my screen better. Okay. All right. So the basal side is where I want to use a double-sided tape. So if you have a double-sided tape, you see that by definition, the tape has a geometry, a very low aspect ratio. So if I can make a channel in that tape, then I won't have to worry about the height. I just have to worry about what's the width of the channel. So what I can do essentially is again a stationary item. So what you can do is this is a slotting machine. So you can place it like that and essentially slot a channel out. Did you see what I did? So I have immediately a channel, right? So just using a slotting machine, and this channel can endure the flow that I'm talking about, okay? Or let's say if you have a unique design that you want to do, then and the slotting machine dimensions are not correct, what you can do is, let me see, you can take your double-sided tape, right? And I'll tell you why the double-sided tape is important, because you want to bond. And as Akshay commented that you guys can see, right? Yeah, commented that bonding is a problem. So the double-sided tape solves one bonding problem, which is bonding to the base. So in the base, we can use glass laminate, a thin glass like this, or plastic laminate, or anything that you can sterilize later. I mean, things that are transparent so that you can see from the bottom of the channel. So you make a rectangle like that, and you can use this to, hold on, too many sharp items. You can make a channel like that. So it's very similar to patterning paper, like the kind of kirigami games that we used to play when we were kids. But it's very useful to make interesting scientific equipments. So once you do that, I'm not doing it properly, but as you can see that I just made a channel. Okay. So this is a microfluidic channel. How? Right now it's kind of a hole, not really a channel. So I can remove one side of the double-sided tape. It's a conductive tape. You don't necessarily need a conductive tape. Oops, there is a problem here. And I can place it on a glass slide, let's say, something like that. Like this. And this is again a tool that kids use to play. Okay, you press it properly, you remove all the air bubbles, and now you have a channel. Okay, a channel which has an aspect ratio of what? I made a 2 millimeter by this, the tape width is 200 microns. So a 2 millimeter by 200 microns, which is essentially 0.1 aspect ratio. So you immediately have a channel which is 0.1 aspect ratio, right? Okay, so now the game is that we want to put a membrane on top of the channel, or we want to make it like my device has multiple wells here. So what I do even actually is something like this. So I add another layer to this double-sided tape, and then I make tiny, there's a lot of glare, I guess, right? Still there's a lot of glare. Channels, tiny compartments like that. So eventually when I place it on the double-sided, on the glass plate, it looks like that. So I place the membrane here. So let's talk about the membrane a little bit. So now we have the basal side ready. Now the question is, where do I get the membrane and what kinds of membrane can I use, right? So at least this part is ready. If you have questions, you can ask. I'll go into the membrane part. Okay. So when it comes to membrane, I use plastic inserts. I usually, let me see. You can also use ECM protein to make your own membranes. The structure is not going to be same. The mechanical strength and other things are going to change, and it gives you some advantages, some disadvantages. But this paper lays out a very straightforward methodology to make your own membranes. So you start with an ECM hydrogel. I mean, many of you have collagen matrigel, right? So you can mix them up in a specific ratio, or you can just do ECM collagen 1. And then dry it out, add water and cross-linking agent. And then you can see that eventually you can peel it out like that. So once you peel it out like that, you can then place it on top of your channel to cover up the bottom channel so that you can place the top channel on the top, right? Or what you can do is you can be a bit lazy and say, well, I'm going to buy a plastic insert. But still, I'm saying this looks like a better approach to me. But let's say that you buy plastic inserts, which are transwell inserts. You cut them open into multiple pieces. So with one transwell, I can make 4 devices. So just to show you, let me do this. No, sorry. It should be from the other side because the basal side needs to go in. And that's where I have kept my permeable membranes. So as you can see, I've just cut them into multiple pieces and I will place them here. I think I should cut them even further. This length should be enough to cover up the channel where I'm going to seed the cells. So this takes a little bit of care and patience when you want to bond. You don't want to touch the location where there will be cells. You just want to bond them, bond the membrane on the side very quickly. Not going to make it look perfect. Okay, so okay, so what I have done essentially is once I have the membrane, I hope you can see the membrane, I have sealed up the basal side. So now there is a channel on the basal side, and on top of the basal channel, there is this membrane. Okay, and then you can access the basal side using these 2 ports, right? So that's basically my device. You can design your own device in whatever way you want. Now, all I have to do is bond these 2 things together. So I have to just place it correctly so that these patterns here, all the 3 wells match these 3 wells here. And then what I'll have is an apical side, or the top where I seed the cells, and a basal side. So I can flush the media on the basal side and apical side and grow the cells. So with that, I would like to go into the next slide. Let me share the screen again. and talk about plasma bonding. Okay, if your entire system is made up of glass and PDMS, let's say, because you, instead of using the double-sided tape, you can spin coat a thin layer of PDMS and make patterns, which is much more difficult to peel and make patterns. So I prefer the double-sided tape. You can also do other things. By the way, this actually came to me just this evening. So what you can do essentially, let's say, to make a mold. So you all know this is a pen eraser, right? So it's something very interesting happens is that this glues very well with with glass when you roll it. So as you can
Iqbal,
see,
just sorry for the interruption, but if you could just stop sharing screen and then do that, it will be clearer.
sorry.
Yeah.
Yeah. So what I did is I just rolled so this is a pen eraser basically. If you write something and you want to erase it and write on top, this is just a die, right? So it's a very thin die. So this dimension is very small in terms of height. So what I just did is I made a pattern here, right? So if I pour PDMS, I take the PDMS out, I immediately have a channel which is around 3 millimeters in diameter and maybe I think less than 100 micron in height, right? So this is also a very frugal way, and this you can sterilize, and this becomes very solid. It doesn't get away. It works very well with glass. So this is another way of making patterns in glass or making molds if you, let's say, don't want to machine. All right. So let's put it back. Let's go back to session 2. Okay. So now, how do we bond these two things that we just developed, that we just fabricated? One is this bottom channel with the membrane, and the other is this top channel. Well, plasma bonding is one way, but plasma machines are very expensive. And the way plasma bonding works, you can always Google how plasma bonding works. You basically change the surface of PDMS and glass to open up certain bonds. You can open up silanol groups and on PDMS and OH groups on glass, and eventually the bond that happens is SiO silicon oxygen and silicon bond, which is fairly strong. Like one comment that I saw is that these bonds are not strong, which I doubt, because when we bond PDMS with glass, you have to break the glass to detach it. It's super strong. But sometimes you don't always have PDMS and glass because you have a double-sided tape here. So what you can do is you can spin coat PDMS, as you can see here, into a thin layer and dip this top in that thin layer. So that will create a very thin layer of glue on the top channel. The layer will be so thin that it won't enter the well, and then you can place it based on how skillful you are. So once you place it there, it will immediately, right now there will be some amount of bonding because it's a double-sided tape, but I don't think this is the perfect bonding that you expect because you don't want any leakages, and we'll talk about leakages after some time. So that's one way. Another way, even cheaper ways, are TV sealant that seals up any, this is basically another form of PDMS, but has a very low curing time. So it cures super fast. So you can use that sealant as well. I have it with me, but it's, you got the idea, right? So one more point right now is that, what about the edges, right? So once it's time to see, wait a minute, did I complete? One more point is that with plasma bonding, you also increase the hydrophilicity of the device and the channel. This is an observation, and then later I read in literature that, yeah, this happens because you can see that the fluid spreads out more. So if you don't plasma treat this, then you will have a hard time flushing fluid initially and also later, because hydraulic resistance will be very high. On top of that, if it is hydrophobic, it won't accept fluid that easily. So you can plasma bond. So plasma bonding helps in bonding and also making the surface hydrophilic. So that really helps. And this is basically a chart of how thin a layer you can make with spin coating. Now, there are interesting questions depending on what your model is. So let's say you want to make a device just for imaging. So it doesn't really matter what happens to the edges. What you want to do is just image this part, right, which is at the center, nice and easy. But if you want to measure transepithelial electrical resistance, TER value, or the barrier strength, or fluid flow, or you want to extract apical and basal side, the molecules and the ions, you want to extract that. So then you need a sealed epithelium, an epithelium that forms perfect bonding on the edges where there are no cells with the PDMS wall or whatever material you use. So in that case, if you have those spatial requirements, I prefer making a device something like this, where the point where you basically look at the barrier or the epithelium does not necessarily include the cells on the edges. So this is just a gross simplification. I mean, you can have a wider channel and have a very tiny channel at the bottom to look at exchanges in fluid, because this part, I mean, whatever be the shear flow, there will always be some non-uniform stresses at the edges, and that might lead to peeling of the monolayer, but that won't harm the information that you are trying to capture in this domain. Okay.
Ikbal, Dhaneshri is asking if PMMA also will work similarly bio method.
Yes, it will. It will. PMMA is it's called acrylic glass, right? So you have to machine it. It's not soft. PDMS has an advantage that it is soft. You just pour it on any pattern and you can replicate the pattern. PMMA is like a plastic, so you have to machine it, and machining will will be expensive because every time you'll have to machine. With PDMS, once you make a mold like this, then you can make n number of devices with it. You don't have to machine it again and again. So let's
just
move
a follow up to that. Just a follow up to that. Is there any other material we could use to make those molds?
Let me think. You can use some gels, but I don't have much experience with those gels. I'm sorry. I mean, polyacrylamide and all. People have made. I mean, one of our collaborators has made microchannels with polyacrylamide gels. But PDMS is like a top choice, and it is not that expensive. I mean, it's everywhere. I mean, there are other polymers like PDMS, but I think people use PDMS because of its biocompatibility. It is much more compatible to cells as compared to other polymers. There's a wide variety of polydimethyl siloxanes which do the same, which have the same feature. They are soft, and you can make them easily by pouring part A, part B, and curing it for some time. You know, I mean, this sealant will behave like PDMS if I pour it on the mold, right? But I'm not sure whether it is biocompatible or not. So I'm just sort of using PDMS for that reason. Okay, I'll quickly jump into the electrical component. So a lot of you want to measure electrical resistance across your monolayer, or let's say you want to apply electric field. So there is an interesting field of investigation where people apply electric field, external electric field, and observe how polarization changes, how proliferation changes. Many people use even TER to do drug testing, because if you have a poor barrier strength, that can lead to, let's say in your guts or in your eyes, that can lead to plenty of diseases. So investigating, so using TER or transepithelial electrical resistance is a very interesting output for many people who are interested in drug development or toxicity development, right? So, okay. So how do we do that in this setup, and how do we do that in a cheap or frugal way? So one method, that you can do, let me, wait a minute, let me unshare the screen. Okay. So the gold standard right now is this device. Okay. So this is a $2,000 device, and basically it's a black box. You don't really understand what's going on. They give you chopstick-like electrodes that you can only use with transwell setups, which are like this, right? Redundant setups. So you can't really use it with the organ-on-a-chips devices. So what you can do is you can what is this? So this is indium tin oxide. Okay, so it's a conductive material that you can coat on a plastic laminate. I hope everyone can see it. So one side is coated with a conductive material. It has very high conductivity. You can buy indium oxide. There are different kinds of conductive ink that you can buy and coat a plastic laminate or glass. So you can easily coat this glass slide with with this conductive material. So now the entire process is same. So now that I want to do electrical measurements, I don't want any double-sided tape. I want a tape that is not conductive, because the only conductive elements that I want are electrodes on the top and on the bottom. So I'll just quickly demonstrate another fabrication. So you have a channel. I'm not going to make an organ on a chip per se with this one. Just quickly wanted to show you what we can do with this. I can create a microfluidic channel with electrodes on the top and bottom. And then, hold on, let me just quickly check which side is the conductive side. Okay. Again, like this. So you have a channel there itself, and top, and you wait for the top, but the top needs to have some ports so that I can add fluid and remove fluid. So I mean, this material cannot be perforated with a blunt tip. So again, I mean, it's funny that I'm using things that are not used in a clean room. So this is a belt puncher, right? So you can, assuming that this is clean, I sterilized everything. I can just quickly make punch holes. Oops. All right. So now I have two clean holes here. Sorry, I'm not doing it neatly because I have limited time, but you get the idea. Okay, so two clean holes, right? And then I have to just place them, making sure that it is aligned with the double-sided tape. So immediately what you have is then you have a microchannel with a conductive top and conductive bottom. You can seed cells here, sterilize it, and we'll talk about sterilization, so don't worry. And then seed cells, you can even reinforce. It seems very flimsy and thin. So what you can do is you can reinforce by punching again PDMS blocks and joining it with this, aligning it here, so that you can have a small tiny reservoir for media when you seed cells and doing all kinds of cool experiments. So that's your small TR measurement tool. You can borrow an oscilloscope from your friend who's in electrical or electronics and just start measuring resistances and also read into the literature and do those experiments. So I'm going to quickly share my screen just to follow up on the electrical measurements. I showed you what you can do with a conductive ink, as you say, as I showed it here. You can also stamp these electrodes using a mold that you make, and then dipping it in like stamping that you see. The other way that I think it's very cool is pattern. So you can stamp it, and the XY resolution is very interesting. So just like this thing here, I mean, this is also for identity protection stamp. So you can make a stamp with a known pattern, and then you can dip it in conductive ink and then just stamp the kind of electrodes that you want to do. So these are like very interesting things that people in maker community work on to make DIY circuits and all. The last part is where I'll talk about sterilization. So you can use UV. Almost all biosafety cabinets have UV. So you can use UV for sterilization. You can use 70% ethanol, flush the apical basal side with ethanol, and then add media. That's one way. If you want to sterilize the entire device, plasma treatment is a very good way. So plasma treatment has many advantages. It is like sterilization, making surfaces hydrophilic, and also bonding, right? So again, this is something that I'm thinking of, and I am trying to collaborate with people to actually make it. So what we can do is that you can, so the idea is plasma treatment requires three things. One is a vacuum, right? So almost all plasma treatment has vacuum. So how do we create vacuum in a frugal way? Like, and this, I borrowed some of the ideas from the frugal science class, is that you can use a 50 ml syringe to create vacuum. So if you block the side of the syringe and pull the plunger, then essentially you get a vacuum. So you can create up to 20 tors and different kinds of, you can connect a vacuum gauge and know what vacuum you have created, right? So you can start with a 12-volt battery. So the next step in plasma development is electric field. So you need electrodes or a strong electric field. And how do you get a voltage of 20 kilovolts? So you can use voltage multipliers that are also easy to get. I mean, toys have voltage multipliers. So you can start with a 12-volt battery and then amplify it to 20 kilovolts and make your own. As you can see here, it's a very frugal way of getting plasma out of almost nothing. So that's one way. I leave you with that. I mean, I have not finished this, but I'm actively trying to get a make a plasma treatment, very frugal plasma treatment machine for myself, and then we'll see who can use it. And this, obviously, I've shown you previously. It is the devices. It is a TIC video of cells growing on the porous membrane in the device, which I made no differently than I showed you today. So the exact same protocol in a much cleaner, neater environment, and then I plasma treat. So the only expensive step that I use in my device fab and before seeding cells is the plasma treatment, which I'm trying to frugalize, if that is the right word. Anyway, with that, I'll stop. I think I've taken a lot of time. If you want to collaborate or want to beta test my prototypes, all the stamps and all, just email me and we can talk about it. Yeah, that's it. And thanks for your attention. Please ask questions.
Yeah, in the interest of time, maybe we can, we'll just take like one question which I can see where Advait is asking, do we need to spin coat it or can we just apply a thin layer to both sides using a brush? Also, do you cure the device after bonding the layers?
You're talking about bonding, right? So if you are using PDMS as a glue between the two blocks here, then you have to cure PDMS. The first step is to make a thin layer of PDMS on one of the substrates and then bond it together. So you have to obviously cure. If you're using RTV sealant, you have to figure out how do you want to make a thin layer. Is it manually? If it is manually, how repeatable that is going to be. If you are spin coating using this thing again, it works and that cures fairly quickly, so you don't have to worry. I mean, an hour is enough to get this device.
Yeah, I think we can wrap it up now as we have kind of exceeded our time.
There's one last question. Yeah. I think there's one other question. Is it possible to mechanically stimulate the membrane in the device and how to do it in an easy way?
Yeah, so as I showed you, hold on. This one, yeah. So if you are using this membrane, which you can make in your lab, this membrane is stretchable. So as you can see that the permeability is a bit low in terms of, I guess, iron and water permeability that they have plotted this. This is not my paper. But the Young's modulus drops significantly. So this membrane is way less stiffer than the membrane that I just showed you. Okay, so you can essentially stretch the monolayer using this membrane if you are interested in monolayer stretching or making a lung on a chip per se.
Yeah, I think that's it.
There are a lot of appreciative comments. Yeah. From Shreyo Sengupta, Dr. Subhankar and Dr. Sandra thanking Ikbal about the wonderful session. Yeah.
Thank you.
So Sham, do you want to give?
Yeah, just that I'll just post a feedback form.
Actually, I would want to form a website where everything is open source and we can form a community where we come up with new ideas to make this. Yeah, everyone can benefit with that from that website. So I'm in the process of making a website where I upload all the items and it will be open source, like an Arduino, right? Everyone can come in and give their input and develop the community accordingly.
Yeah. So just another thing, I've just posted a feedback form in the chat box. So if people could just go in and fill the details. Apart from it, we are very thankful to Ikbal, who has taken his time out and showed us very interesting ways of making a like an organ on a chip, and also for his interesting presentation and insights into how to design it using frugal methods. We are very thankful to Ikbal for giving us this presentation, and also we are also grateful to all the participants who have stayed this long and made sure they got all the information out of him. So yeah,
Yeah,
thank you
thank
all.
you. If you have questions,
Yeah.
you can email me. And thanks a lot, Surat and Vrinda and everyone for organizing this. Yeah.
Thank
Yeah,
you,
we
Ikbal.
hope that.
Uh,
Yeah, Brinda.
Thank you, everyone.
Yeah, also we'll be posting this session on YouTube, on our YouTube channel and Biomed 21 website. So, and we'll be sending the links soon to everyone who wants to revisit this wonderful session.
Yeah.
Yes. Yeah. Let me know. I mean, where you post it. Yeah.
Sure,
Sure. We'll email you all the details of that.
Thank you.
Thank you.
Thank you all.
Again, good morning, everyone. I'm gonna crash now. It's already 2,
Yeah.
2 a.m.
Bye.
Yeah, you look exhausted. Thank you.
Okay. Bye.
Bye.
Bye.